Vanessa. Good afternoon, everyone. It's great to be here. I am excited to share a recent study we conducted looking at sex-specific associations between traumatic brain injury and brain structure. So a little bit of background on what led us here today. So a lot of this we know. We know that dementia is a growing epidemic. 57 million people are diagnosed today, and this is predicted to exceed 150 million diagnoses by the year 2050. So a lot of work has been done on modifiable risk factors. We've spoken a lot about them today. The ApoE4 gene is the strongest known genetic risk factor for dementia. But another non-modifiable risk factor is actually sex. So sex is considered a non-modifiable risk factor, and this is because females account for up to 2/3 of all dementia cases globally. So a lot of work is being done in various areas, looking into why females might be at a higher risk for dementia. And one of these areas that they're looking at is around sex hormones. So, which was importantly brought up earlier, estrogen, one of our sex hormones, is possibly neuroprotective. So what that means is it kind of enhances neural plasticity and it can reduce neural inflammation. So when women go through hormonal transitions, such as the menopause, they see a decline in these sex hormones. And it's thought that the decline in these sex hormones may make women more vulnerable to neurodegeneration. But a lot more work is needed in this area to better understand the mechanism behind it. So, you've seen this graphic many times today, but I'll touch a little bit on modifiable risk factors for dementia. But I'll draw your attention to one in particular. So traumatic brain injury is considered a modifiable risk factor for dementia. This is because there's evidence to suggest that it causes both cognitive and structural impairment, and this can occur in both acute and long-term stages after injury. So the Lancet Commission on Dementia has also said that it's possible that TBI leads to earlier onset of dementia by up to 2 or 3 years. Now, interestingly, women are reported to have worse outcomes following TBI. So women consistently report greater TBI symptom burden and worse functional outcomes following TBI. This is pretty consistent in the research on mild TBI. However, the research on severe TBI is a lot less consistent. So we wanted to address a few of these gaps. So we wanted to look at how does severe TBI impact cognition in midlife specifically, and we wanted to have a particular lens looking at sex differences. So we wanted to see how does severe TBI impact cognition for females, and if this differs between females and males. And we also wanted to have a look at whether genetic and hormonal factors played a role. We wanted to do the same thing for brain structure. So we looked at whether or how, I suppose, severe TBI impacts brain structure in midlife. And again, we looked at whether this differs between males and females and whether genetic and hormonal factors were at play. And just a note here, in the context of this study, severe TBI was any TBI with loss of consciousness. Hormonal factors, so the women of our cohort were stratified by their menopausal stage. So women were either premenopausal, perimenopausal, or postmenopausal. And then all participants were stratified by their ApoE4 genotyping as either being a carrier for the gene or a non-carrier for the gene. So what did we find? Well, our participants were from the Prevent Dementia study. So it's the world's largest longitudinal study investigating dementia risk factors in midlife. We had 448 participants in this analysis. Males and females were well matched in terms of demographics and genetic factors. However, we found that males sustained significantly more severe TBIs than females. So what do we find in terms of cognition across the entire sample? Well, we didn't see a significant difference between males and females on how TBI impacted their cognition. However, we did find that severe TBI was significantly associated with worse cognition across the entire sample. In our subgroup analyses on females, we observed that postmenopausal women performed significantly worse on cognitive measures compared to pre- and perimenopausal women. However, there was no interaction between TBI, menopausal stage, or ApoE. So looking at some brain structural analyses, across the entire sample, we did not find evidence of an association between severe TBI and total grey matter volume within the brain. However, our subgroup analyses on the females, we observed a really interesting finding. So we actually found a significant three-way interaction between TBI, postmenopause, and ApoE4. Specifically, Postmenopausal women carrying the ApoE4 gene showed significantly greater grey matter volume loss with TBI compared to non-carriers of the ApoE gene. So it suggests that it's possible that women who have gone through menopause, or who are postmenopausal, I should say, who have seen a decline in estrogen, this neuroprotective hormone, and who are carrying the ApoE4 gene may be at higher risk for neurodegeneration or structural impairment following TBI. So just to summarize what all this could mean together. In terms of cognition, we did find evidence to suggest that severe TBI is significantly associated with lower cognition across the entire sample in midlife. Postmenopausal women perform worse on cognitive measures than pre- or perimenopausal women, and this is after controlling for age. So it suggests it's possible that there are cognitive changes occurring in postmenopausal women, irrespective of the aging process. However, there were no interactions between severe TBI, menopause, or ApoE4 to impact cognition in midlife. Regarding brain structure, we found that men in this cohort sustained significantly more severe TBIs. However, women are still showing a greater impairment following severe TBI. So again, it's more evidence to suggest that women are experiencing worse outcomes following TBI compared to men. Now, severe TBI was not associated with total grey matter volume in our study. This is actually in contrary to previous research. There has been evidence that's found a link between severe TBI and grey matter volume loss in the brain. It's possible that with our midlife cohort that they're just too young for structural impairment from TBI to have manifested yet. So a lot of TBI research looks at midlife to older age adults, so the average age tends to be a little bit older. So I think this study, being longitudinal, is well placed for us to follow them over time, see if and what patterns of brain volume loss may emerge following TBI. And lastly, our main and novel finding is that postmenopausal women carrying the ApoE4 gene showed significantly greater brain volume loss, grey matter volume loss with TBI compared to non-carriers. So it suggests this group might have heightened vulnerability. The genetic risk may interact with postmenopausal changes to accelerate volume loss in the brain following TBI. So I think it really highlights the importance of considering both genetic factors and sex-specific factors in TBI outcomes. And again, I think it'll be imperative for us to follow these participants over time to see how these patterns continue long term. So a special thank you to the PREVENT team for their dedicated work on this longitudinal study and to my supervisors for overseeing this work.